Okay, I think we're good to go.
All right. Should I just start? Do they need an introduction?
You
I'm
can,
from MIT.
well, yeah, tell them a little bit about yourself.
Yeah, okay. I'm a senior research scientist at MIT, and I've been working on toxic chemicals and disease since about 2007. I've published many papers, over 30 papers in peer-reviewed journals. Super excited about deuterium right now. It's just totally grabbed my heart, and I am so into it, and you will appreciate it from this talk. And this paper, that I'll be focusing on a paper that I think Martha has seen. It's a preprint, but I'm happy to say that the reviewers liked it, and we've submitted already the revised version. So I'm very hopeful it's going to become published in a peer-reviewed journal. So, here's my topic: the role of deuterium and glyphosate in Parkinson's disease. And here's an outline first, an introduction. Then I'm going to talk about the gut microbes and how they supply deuterium depleted nutrients to the host. Very, very important and very significant aspect of human health. I'm going to talk about the role of histamine and mast cell activation in Parkinson's disease. And then I'm going to talk about cardiolipin, a very interesting lipid that shows up in the mitochondria. And I think that it is able to actually trap deuterium. So I'm going to get into some very interesting physics and biology and hypotheses. A lot of it's hypotheses not quite proven yet, but I'm hoping that somebody will do the research necessary to show that there's something to it. So deuterium, people probably may not know what deuterium is. It's a natural heavy isotope of hydrogen. So hydrogen is the smallest atom in the periodic chart, and deuterium has got an extra neutron, which makes it twice as heavy. It's a natural element. Although it's rare compared to hydrogen, it's common because hydrogen is so common. It's 5 to 6 times the concentration of calcium in the blood. The critical thing about it is that it breaks the ATPase pumps in the mitochondria, which causes energy loss, as they can't make as much ATP, and also generates reactive oxygen, which then causes all kinds of trouble to the cell. And of course, defective mitochondria is a core component of many chronic diseases. NAD, nicotinamide adenine dinucleotide, it carries an H, and that H is usually depleted, very low in deuterium. This whole mechanism that tries to keep deuterium out of NADH, and then it delivers that H to the mitochondrial intramembrane space to drive these ATPase pumps. Very important point here. So the mucins in the gut are sulfated, and the sulfation causes gelled water, and the gelled water releases protons that are probably not deuterons. I think that's a crucial way in which you can deplete the amount of deuterium that's in the fluid water, in the lumen, where all the activity is happening with the gut microbes. And so that happens also along all the blood vessels. They have sulfated glycosaminoglycans that I believe are trapping deuterium to keep deuterium out of the flowing blood. Glyphosate suppresses many proteins involved in deuterium homeostasis. So this is a very big picture, important points on that slide. So there's a paper that I found in The Lancet from 2023, and I was unhappy to see this because it shows that it's suspecting glyphosate is a causal factor in Parkinson's disease. And it pointed out that it's the fastest rising disease among the neurological diseases worldwide. And pesticides can cause neurodegeneration by affecting the gut microbiome, which is really the big topic of this talk. Neurodegenerative processes spread from the intestinal neurons via the vagus nerve to the brain. And then there are multiple pesticides that are synergistically toxic to dopaminergic neurons, which are, of course, the critical ones in Parkinson's disease. Glyphosate can cause, and there's many papers that have shown that glyphosate causes mitochondrial stress, oxidative stress, neuroinflammation, mitochondrial dysfunction. Independent studies should be part of a regulatory risk evaluation process. So they're criticizing the regulators for not finding more about this issue of glyphosate potentially causing Parkinson's disease. So this is a not peer-reviewed paper. This is the one I just mentioned on Parkinson's disease. It's a preprint. It's available on the web as a preprint. And I'm very hopeful that it's going to get published soon. So mitochondrial dysfunction in Parkinson's disease is impaired deuterium-depleted nutrient supply by gut microbes, a primary factor. And this is going to pretty much be the focus of this talk. I want to show this. This is kind of an outline of what's in that paper and the steps that go on in finally producing Parkinsonian symptoms. So you start with an unhealthy gut microbiome. Of course, glyphosate causes that. Then you don't have an adequate supply of butyrate, which is a low deuterium nutrient for the colonocytes. This causes mitochondrial dysfunction, and in enteric neurons in particular get dysfunctional mitochondria, which causes alpha-synuclein misfolding in the gut. And then that gets transported via the vagus nerve to the brain, where it sort of triggers further misfolding of other alpha-synuclein molecules that are there, causing lipid sequestration in lipid bodies, which I consider to be deuterium trapping, and I'll get into more details later on. And then it causes an impairment of these dopaminergic neurons, which need a lot of ATP, and finally you get the Parkinson's symptoms. This is my book, Toxic Legacy, How the Weed Killer Glyphosate is Destroying Our Health and the Environment. It was released by Chelsea Green in 2021. It was almost a decade of work led into finally writing this book. I have lots and lots of references in there, extensive data on evidence of toxicity to animals and humans. And in particular, I show how it interferes with sulfate homeostasis, which is going to matter with respect to trapping deuterium in gelled water, because sulfate gels the water that lines the gut and that lines the blood vessels. And this book argues the very important point here. It argues that glyphosate is insidiously, cumulatively toxic through its diabolical insertion into proteins by mistake in place of the coding amino acid glycine. I think this is extremely important. It's not proven. In fact, it's quite controversial. The industry is denying that it's possible. But if you read my chapter in the book where I explain it, it looks to me like it's almost, the industry has almost proved that it's happening. I believe the evidence is very strong. This book was selected by Kirkus Reviews as one of the top 100 nonfiction books of 2021. So my next topic, gut microbes supply deuterium-depleted nutrients to the host. Here's the big picture. The gut microbes produce hydrogen gas, H2, and it's remarkably depleted in deuterium. This gas is recycled back into organic nutrients by other microbes, and those nutrients then are also deuterium depleted. The enzymes that are involved in this process have strong dependencies on glycine residues that could be substituted by glyphosate. And I think this is what's causing, we have sort of an epidemic and issues with the gut, bloating, gas pain. They could be caused by impaired hydrogen recycling due to glyphosate exposure. Butyrate is a short-chain fatty acid, and it's sourced from hydrogen gas, which means that it's going to be low in deuterium. It suppresses mast cell activation and histamine release, which are associated with Parkinson's disease. Hydrogen-enriched water and other hydrogen therapies have shown therapeutic promise. The benefit might be because that hydrogen gas is low in deuterium. This is a fantastic paper from 1961. I'm sorry to say that I haven't found anything more recent than this paper, but this one was really good. Experimentally, they showed, they looked at a microbe producing hydrogen gas, and they discovered that it was only about 30 parts per million, whereas in seawater, it's 155 parts per million. So this is like 80% down on the deuterium in the gas. And this is probably partly because deuterium tends to stay with a more solid state, from ice to gelled water, to fluid water, to gas. In each step of the way, deuterium stays behind. So you get depletion of the gas, in the gas. And they use both glucose and formate as substrates. To the enzyme yanks the hydrogens off of these molecules and produces hydrogen gas. And they showed that the water that was left behind did not have this depletion. It was only in the gas, which is important. So we know in the gut that the bacteria do a lot of fancy things. And the fermentation process involves breaking down small organic molecules, extracting the hydrogen. It's sort of anaerobic processing. You extract hydrogen. You make hydrogen instead of water. You don't use oxygen. You make hydrogen gas plus carbon dioxide. And then you can put those back together again to make very important nutrients that supply the host. And there's 3 different kinds of classes of bacteria that will take that hydrogen gas, use it as a reducing agent. And you can get methionine, which has this universal methyl donor, CH3. These are going to be low in deuterium. Methane, which eventually becomes NADH, this is going to be low in deuterium. And then the acetogenic bacteria might be the most important ones. They produce acetate, which then becomes butyrate. Acetyl coenzyme A is the fuel for the mitochondria. That's what the mitochondria break down to produce carbon dioxide and water in the oxidative phosphorylation and metabolism, citric acid and the oxidative phosphorylation process. So this is just showing this universal methyl donor. This supplies DDW to the mitochondria, and this is the citric acid cycle, which fuels the ATP pumps. So you can see those are all super, super important in biology. Now, these BTBR mice are mice that are used experimentally as a model, a mouse model of autism, because they have autistic behaviors. They breed true to that, which is quite interesting. So they use them in studies on autism. This paper showed that they had reduced levels of acetate in the gut. And what they did was they exposed these mice to glyphosate at different levels. These colors are all the amount of glyphosate exposure, the red, blue, green, and this was unexposed. But all of them had glyphosate because it was in the food as well. So even the unexposed came up with measurable levels of glyphosate. But there's a trend line going up like this and going down over here with a very significant p-value, 0.0001 in both cases, glyphosate and pH, and glyphosate and acetate. So what happens is these bacteria that make the acetate are like a low pH. And glyphosate raises the pH of the gut, which messes things up. Those microbes don't thrive. They don't make enough acetate. They don't make enough butyrate, and you become butyrate deficient. So I think this is what's going on with glyphosate. And the children with autism also have reduced amounts of acetate in their stool samples compared to controls, 5.1 versus 3.5. So this is a schematic that I drew to sort of trace the path to butyrate, starting with dietary fiber. And you can have these milk products, HMOs, inulin, starch, pectin, all these fruit starch, various kinds of fiber. They get broken down by these primary degraders, like bifidobacteria and lactobacillus, into small organic molecules like glucose and fructose. And then the secondary degraders are the ones who break these down further, make the hydrogen gas, and then turn it into acetate, propionate, and butyrate. These are the 3 short-chain fatty acids. And the butyrate in particular is something the mitochondria in the colonocytes love. That's their favorite food. If you feed them a lot of butyrate, they're healthy. And they can use it to turn it back into acetylcoenzyme A, run it through the TCA cycle, and then eventually produce DDW with the production of ATP. So butyrate is probably very depleted, and it's the favorite fuel source of the colonocytes lining the colon. Bifidobacteria, there's lots and lots of papers coming out on them and showing how crucial they are for the health of the gut. This paper talks about a beneficial role in colitis, a mouse model. When they provided them with bifidobacteria bifidum supplements, their colitis improved. They had reduced inflammation, reduced levels of neutrophils, which are immune cells, and enhanced tight junction health. So all kinds of good things. It also inhibits the growth of pathogens and increases the production of the short chain fatty acids, such as butyrate. Such as butyrate. And now it turns out bifido and lactobacillus are the 2 bacteria that have been singled out as being most sensitive to glyphosate. So they get killed off by glyphosate, and that allows other microbes that could be pathogens to thrive. Here's another paper on the benefits of bifidobacteria, and it talks about other species as well to end up with the butyrate. The bifidos turn the starch into these small molecules. Firmicutes and bacteroidetes produce the hydrogen gas, and then firmicutes produce butyrate from the hydrogen gas. And then all the species need to be abundant to support hydrogen recycling. That's a very important point. It is said that approximately 10 liters of hydrogen are produced daily in the human intestines. Low hydrogen levels in the breath are linked to Parkinson's disease, rheumatoid arthritis, cardiovascular disease, and Crohn's disease. So next topic, histamine and mast cell activation in Parkinson's disease. Here's the big picture. Mast cells release histamines in response to allergens. Many people have a problem with mast cell activation syndrome. Histamine contains an imidazole ring. This is a very interesting physical ring with very interesting characteristics in that it can trap and retain deuterium. Histamines are metabolized primarily by an enzyme called histaminase in the gut. Histamine sensitivity results when histaminase is defective. When it's suppressed, and I will argue that glyphosate suppresses it. A large number of uncomfortable symptoms arise from histamine intolerance. Glyphosate likely suppresses it. My hypothesis is that mast cell activation is induced when the gut lumen contains too much deuterium, which could be due to insufficient butyrate and insufficient sulfate in the sulfomucins. And also a hypothesis is that the metabolites of histamine can trap deuterium and permanently sequester it. So this is a paper on mast cell activation in Parkinson's disease. It's actually associated with Parkinson's. Mast cells are more abundant in the PD brain than in healthy controls. Dying neurons promote mast cell recruitment. And then mast cell activation then causes neuroinflammation. Mast cell degranulation activates microglia, which exacerbates the blood barrier disruption. And there's a little picture here. This is the alpha-synuclein, microglia, astrocyte. This is the, sorry, I'm a little confused on who's who here, but anyway, I think these are probably the mast cells. The M is the mast cells that are causing a neural death with releasing all of these different things, including histamine here. This is mast cells, these are microglia. So this is what is amazing to me. I always rummage through the web looking for proteins that have what I call a glyphosate susceptibility motif. And that motif is defined as having at least one glycine residue at a site where it binds phosphate. And it turns out there are many proteins that have this property, and many of them are involved in deuterium management. So histaminase has a fantastic motif, about the best I've ever seen. This is the motif here, YGHGGGWG. These G's are glycines. So it's got 1, 2, 3, 4, 5 glycines in this motif. And this and the enzyme matches it perfectly. This is the actual sequence in the enzyme. And this is a lovely plot that you can get on the web from a fantastic service where it shows you exactly where things happen. This is FAD, which has FAD has these two phosphates, and it binds those two phosphates. It's the P's here. It binds those phosphates right here where there's these 3 glycines. So it's a perfect setup for glyphosate's mischief. And I believe that glyphosate is likely suppressing it. This is a paper that I published with Magnai and Anthony Kirikopoulos. Notice that it was published on May 14th, 2025, so just a few days ago. I'm excited to get that out. And this is deuterium sequestering by reactive carbon atoms, an important mechanism to reduce deuterium content in biological water. That's the title. In the abstract, specifically, we've identified the imidazole ring, and that's what's present in histidine and in histamine, and the microderivative of histidine, such as imidazole propionate. And then there's also these other molecules that all have characteristic, uniquely situated carbon atoms that can trap deuterium. And also these bisyllabic carbon atoms, which I'll get to in a moment, in the polyunsaturated fatty acids. All of these molecules can likely trap deuterium and hang on to it for dear life and not let go. And so they could sequester deuterium and thereby reduce the deuterium levels in the water-based medium. That's sort of the proposal of that paper. This is a picture of an imidazole ring in a molecule right here. It's a ring with 2 nitrogens and a carbon in the middle. This is the carbon that's very, very unusual. It has a proton attached to it. And it can, if you put it into heavy water, it's the only carbon atom in the whole protein or peptide sequence that will actually take up deuterium. None of the other carbons are willing to grab it because the carbon really hangs on tight to its proton usually. But all the oxygens and nitrogens all pick up deuterium right away in heavy water. But when you put it back to light water, regular water, in an acidic condition, these oxygen and nitrogen atoms immediately lose their deuterium. They give it up again. They don't hold on to it. Whereas this carbon here holds on for dear life and won't let go. Even in these extremely conducive environments to let go, it doesn't do it. So it's really interesting that it grabs deuterium and then it doesn't let go, which is kind of a perfect setup for trapping it. It's what I suspect happens. So this was an interesting paper that I found looking at different molecules, organic molecules that contain imidazole rings. Histamine right here is the red. And this is the level of activity of this substitution of deuterium for hydrogen going up as a function of pH. And so when the pH gets higher, these other molecules, IPA and acetylhistidine hydroxyl, these 3 molecules who also contain the imidazole ring become more active. But especially this IPA is a fantastic example of an ability to trap deuterium. It has a much higher reaction rate to trap deuterium than the other ones, especially when the pH is high, IPA. So this is imidazole propionate, propionic acid, if you will, that's made by gut microbes under conditions that are defective. In fact, under high basic conditions where it can then trap this deuterium. So my hypothesis is that histamine or histidine metabolites can trap deuterium when the gut lumen becomes too basic, the pH is too high. They essentially trap and remove deuterium landmines. That's what I talked about in that paper that we saw. Imidazole propionate. So that's an interesting molecule, IPA. It's produced by gut microbes when the pH is too high, and its presence is strongly associated with diabetes. So I find that really fascinating because I think diabetes is caused by too much deuterium, like I think everything is caused by. And so IPA is able to trap those deuterium atoms and improve the situation. So these are the different molecules here showing the pKa. This imidazole propionate has a very high pKa up here at 7.6. And when you have a high pKa, you might have a much higher reaction rate for trapping deuterium. So you want to have a molecule that has a high pKa, that has an imidazole ring with a high pKa in a basic environment, which is perfect for this imidazole propionate in association with diabetes, to then trap the deuterium and not let go. So this is sort of my summary of what's going on. Well, this is a paper that talks about a theory which completely matches my theory with regard to glyphosate. They don't mention glyphosate, but it makes it very clear. This is the enzyme that produces IPA. It's active at neutral pH, but the colon tends to be acidic. So if the colon pH is too high, which could result from decreased production of short-chain fatty acids, such as butyrate, and or increased protein fermentation in the gut, both of these things are caused by glyphosate. And so that could increase the production of this to cause a diabetes, and diabetes is going up dramatically, exactly in step with glyphosate usage. So it causes the increased pH, as we already saw, and it also increases the production of ammonia. I wrote about that in my book. I have a chapter on the gut where I showed that glyphosate disrupts protein metabolism, so you end up with peptides making their way into the colon and getting fully metabolized to ammonia, as opposed to being absorbed as amino acids, because it's too late to absorb them once it gets into the colon. So you end up with a high pH, and ammonia can also be problematic. So mast cell activation syndrome was first recognized as a disease in 2007. It's associated with excessive inflammation and allergic reactions. Mast cells release abundant histamines into the gut lumen. Histamines act as signaling molecules to induce diarrhea, abdominal pain, and nausea. Histaminase metabolizes histamines into metabolic products that might be able to trap deuterium and remove it from the gut. Suppressed histaminase causes histamine intolerance. Histaminase has a very strong glyphosate susceptibility motif. And so the gut microbes metabolize histidine into this imidazole propionate, and elevated levels are linked to diabetes. IPA is very good at trapping deuterium. So glyphosate induces an environment in the gut that is conducive to IPA producing bacteria, and that's also linked to diabetes. Okay, my last big topic, cardiolipin and deuterated short-chain fatty acids. Here's the big picture. Cardiolipin is a unique phospholipid with 4 PRUPAs bound instead of the normal 2, and it's synthesized in the mitochondria. It's intimately connected with critical proteins in the inner membrane of the inner membrane of the intermembrane space of the mitochondria. Is a bit of a mouthful, and it is essential for oxidative phosphorylation. And especially in neurons, it preferentially accumulates DHA, which has 5 of these bisallylic carbonate atoms, and these are what I call deuterium traps. And I wrote about this in that paper that I referenced. Cardiolipin accelerates the misfolding of alpha-synuclein, and the Lewy bodies that form in association with Parkinson's disease contain a core of lipids from mitochondrial debris. So I think that the lipids are gathering up deuterium and then getting trapped in the Lewy bodies. That's a way to sequester deuterium away from the mitochondria. That's what I'm proposing in the paper and in my research. Okay, so here's some pictures of some fatty acids with these bisallylic carbon atoms. They're very interesting. These arrows point to the bisallylic carbons. This is a carbon chain in a fatty acid. This is EPA. This is DHA. These are the highly polyunsaturated PUFAs. Arachidonic acid has 3, and linoleic has only 1. So all of these have bisallylic carbons that are linked by single bond linked to 2 carbons that have double bonds. You can see there's double bond, single bond, double bond. That's the pattern all the way through here. And these carbons are very reactive. They cause a lot of trouble in the membrane with reactive oxygen, but they also, I think, can trap deuterium. And I think that that might be the whole reason why all of that stuff happens, that causes a lot of damage in situations where deuterium is problematic. So the deuterated DHA protects the retinal pigment epithelial cells. This is a paper from lipid peroxidation by quenching the chain reaction. Really fascinating that what they are finding, they can actually supplement with DHA that's already deuterated, prepare it in the lab, and take the deuterated DHA. It goes into the retina and protects from reactive oxygen by quenching the chain reaction that normally happens in lipids when they're exposed to oxidative stress. So this is kind of a schematic of it. I kind of like this as an illustration of the situation. Matchsticks in a line with 1 removed. It's a perfect metaphor for the effect of deuteration on bisallylic carbons, where once it's deuterated, it no longer reacts and the reaction chain gets broken. And so these are all lined up, and normally they just go down like dominoes. But this can stop the reaction and actually greatly reduce the damaging consequences of the oxidation. If there's enough deuterium in just some of the lipids, the reaction rate goes way down. This is an absolutely fascinating paper. I recommend you reading it on kinetic isotope effects during the oxidation, oxygenation of polyunsaturated fatty acids. What they did was they had all kinds of different versions of deuterated arachidonic acid. This one has 3 of these bisallylic carbons. They put deuterium on various ones of those bisallylic carbons in this molecule, and they saw what happened if they do that. They found C10 especially important. This particular carbon in arachidonic acid, if you put deuterium there instead of hydrogen, it totally changes the reaction rates because of the deuterium kinetic isotope effect. And all of a sudden, the cyclooxygenase shuts down. The COX enzyme shut down, And so it goes towards the LOX enzymes instead, which actually produce resolvins, which can actually solve the problem of inflammation. So it goes through a process of causing inflammation, but then eventually picking up enough deuterium to solve the problem, and then to resolve the inflammation and to heal the joint or whatever it is that's inflamed. So in simple words, arachidonic acid can pick up deuterium from the medium at a bisalylic carbon atom. And when it does this, the enzymes that produce inflammation-inducing metabolites during lipid peroxidation shut down. And this allows a redirect to a beneficial pathway that produces resolvins, which are the lipid products that tame the inflammatory response. So I think this whole process may go on everywhere in the body where there's inflammation. And the whole point of the inflammation, in a way, might be to gather up those deuterium atoms in those lipids. So this was an interesting paper that actually, again, supplied deuterium-loaded linoleic acid. It has only one bisalylyl carbon, but it gave it both 2 deuteriums instead of 2 hydrogens on that carbon. And they found that it actually goes into membranes. It preferentially goes into the cardiolipin and into the precursor of cardiolipin, which is this phosphatidylglycerol. So these were particularly attracted to receiving deuterated lipids. So I'm imagining that any time a lipid gets deuterated through all this reactivity, it can actually make its way into cardiolipin to help protect the mitochondria from oxidative stress. So they are enriched in PUFA and exclusively located in mitochondrial membranes. And this represents major targets for lipid peroxidation, which of course is very damaging. And of course, mitochondria produce a lot of reactive oxygen. So it seems crazy to put something there that is so sensitive to oxidation damage. But yet, if it can trap the deuterium, that may be more important, and maybe the important role that it plays in the mitochondria. So it can preserve mitochondrial function under oxidative stress. So this is so it turns out cardiolipin is completely, the ATPase pumps are surrounded by, they're enmeshed in cardiolipin. They're soaking in it, in that membrane. And they basically, it's embedded in those pumps, which serves to trap and buffer protons. So this paper talks about trapping and buffering protons, that the protons actually accumulate inside these lipids and become a buffer to keep the proton flow going. But I suspect it may also trap and hold deuterium. So my thought is that it's a filter. It's like a water filter that filters out the deuterium and lets the protons go through. This is theoretical. It hasn't been proven, but I think it's a very interesting thought, and it would make a lot of sense to explain what's going on in the mitochondria. So my hypothesis is that cardiolipin allows protons to propagate through the lipid chain to arrive at the ATPase pump, but it traps the deuterons at these bisalylyl carbon atoms, thus protecting the membrane lipids from peroxidation damage. And then cardiolipin promotes pore-forming activity. This is so fascinating to me. In fact, it turns out that it's not just alpha-synuclein, but many other proteins that can misfold and cause various kinds of fibrous accumulations of basically prion-like diseases. Many of these proteins get triggered by cardiolipin to misfold, and actually to misfold in such a way as to form a pore in the membrane. This turns out not just true of alpha-synuclein, but many other of these molecules do the same thing. They form a pore in the membrane, and the membrane has a lot of cardiolipin. And so it actually cardiolipin migrates to the outer membrane after it's become oxidized, and that presumably after it's caught deuterium atoms. And then in the outer membrane, it attracts these proteins like alpha-synuclein, and it causes the alpha-synuclein to enter the membrane, which folds into a structure that enters the membrane and forms a pore, which then, of course, destroys the mitochondrion, causes mitochondrial swelling and cytochrome C release, and basically could even kill the entire cell. So it leads to organelle swelling and release of cytochrome C, and this can launch an apoptotic death cascade. So lipids are sequestered in Lewy bodies. That's another paper that I found, and this is exactly what I was expecting to find, which is that, and this, don't worry about all these, I'm not going to explain about all these different enzymes here, but basically the mitochondrion is dysfunctional because it's got too much deuterium. It causes oxidative stress. This causes alpha-synuclein to misfold. The lipids accumulate because autophagy fails. And then these lipids that are accumulating, I think, have trapped deuterium. And then the lipids go into the Lewy bodies along with the alpha-synuclein. So you end up with mitochondrial membrane debris that are found inside these Lewy bodies. and that probably contain high levels of deuterium that were trapped while that cardiolipin was living on the inner membrane. And so in summary, gut microbes strip deuterium from organic molecules by recycling hydrogen gas. Glyphosate in Roundup disrupts the gut microbiome, interfering with the production of short-chain fatty acids. Parkinson's disease begins in the gut and is associated with reduced levels of butyrate, which is a depleted short-chain fatty acid. Histamine and histidine metabolites may be able to trap deuterium. Cardiolipin may protect the ATPase pumps from deuterium overload. And my hypothesis is that cardiolipin can seed alpha-synuclein misfolding to sequester deuterium in Lewy bodies, reducing deuterium exposure to the mitochondria. So when you're under deuterium stress, this can all happen to help to keep the deuterium away from the mitochondria. An organic diet rich in low deuterium nutrients may protect from Parkinson's disease. And so we're done. I can stop.
Thank you.
And I hope you could hear that okay. I tried to shout, but
Thank you. Well, we'll take a few questions, but I want to start maybe with just a point. Or so I had been looking at cardiolipin as there's elevated antibodies to cardiolipin in Parkinson's, which maybe, I don't know if you're aware of this, but also there are a number of microbes, pathogens in particular, that actually make a cardiolipin that is very similar to our cardiolipin. So there's a connection there. I'm not sure what it is, but I put that out there for you.
That's very interesting.
So Daniel has a question. I'll have to repeat it, but so well, I don't know. Yeah, we'll put it this way, and maybe you can see the room. Can you see the room?
Yeah, I see you. I hope you can hear me okay.
So I'll repeat the questions. Daniel, what's your question?
Yeah, I have basically two questions.
So 2 questions.
So one is like this question will recapitulate what you said, what you talked about, but what is your take on the chronic use of hydrogen pump inhibitors like omeprazole and chronic use of.
So what is your take on the chronic use of hydrogen pump inhibitors like the PPIs,
Nexium, right?
and
Nexium,
chronic use of antihistamines?
Prilosec and Nexium.
chronic use of antihistaminics?
Oh, antihistamines. Oh, I see. Yeah, well, I mean, because histamines are so sensitive, if you're so sensitive to histamines, you sort of want to stop all those symptoms. It's not fixing the problem, obviously. I mean, it's just blocking the ability. It's problematic when histaminase doesn't work. And I think the only way to fix it is to fix histaminase. And the way to fix it is to stop being exposed to glyphosate and maybe other things that disrupt it too. But, you know, I think a lot of these get-arounds are not really solving the problem. They're just kicking the can down the road, so to speak.
Has she found that fulvic and humic acids are a good way to push back the impacts of glyphosate?
So what have you found that humic and fulvic acid?
Yes, a lot of my naturopath friends have pushed that concept of taking fulvic acid and humic acid, also bentonite clay. These are binders, and the humic acid may even have enzymes in it. Often there are enzymes that are trapped in there that are very versatile, and they might be able to break down the glyphosate. That's theoretical. But I'm always looking for some way of having enzymes that break it down. If you've got microbes living in your gut that can metabolize glyphosate, that's awesome, because it actually has a nitrogen and a phosphorus atom that can be useful nutrients if you can break it down. But it stumps almost all species. It has this difficult carbon phosphorus bond. And most species don't know what to do with that. So that's why it sticks around and causes a lot of trouble.
Don Huber gets all the credit. Holland.
Hi, Stephanie. That was a wonderful talk. Thank you so much.
Thank you.
Indiana University. I love your mast cell hypothesis. We have some single cell proteomics and transcriptomic data on mast cell and plasma cell involvement. What kind of data would you like to see as a proponent of the hypothesis that mast cell biology is important? What kind of data would you like to see in a Parkinson's colonic biopsy to interpret that as a support or rejection of the hypothesis of their involvement?
Oh boy, I'm not sure I can answer that. I mean, I think it would be very interesting to look at things like the level of deuterium in the feces, you know, because I would predict, I mean, in my papers, I talk about even bile acids. Bile acids get metabolized into, I forgot the name of it. There's a complicated name for a metabolite that the microbes make out of bile acids that actually, not bile acids, but the heme breakdown products that are in bile acids. Forgotten the name. It's got a long, complicated name that comes out of bilirubin and biliverdin, and then something I can't do it. my brain has it, but it won't come out.
I'm a bile acid expert in my group, so that's great that you think those are relevant.
Yeah. So I think if I believe there are molecules in the gut that trap deuterium, and that then you could see it in the feces. I would think that the deuterium levels in the feces ought to be high. That'd be the first thing that I would be curious about in general, you know, and even in healthy people, because that's an indicator that you've got healthy deuterium traps. But then, of course, if you've got enough sulfomucins, you might not need it. So I'm not sure how to interpret whatever you might see. But
okay,
yeah.
thank you.
Other questions?
So, well, I have another, I don't know if it's a question or, but I think maybe we connected years ago through Laszlo or, and
That's
one
likely.
One of the things, so Laszlo Boros, if you're not familiar, he's one of the kind of pioneers in the whole field of deuterium. And I had reached out to Laszlo about writing a paper on the probiotic I developed for my husband for Parkinson's, because the formula, so I made a formula to produce mannitol in the gut based on the mannitol research that had shown that mannitol could stop the aggregation of the proteins. So I was like, can we put a factory back? And so I asked Laszlo to look at it, and he went through it and did a presentation for me. And I said, can you write a paper on this? Because essentially the bacteria in the gut, in the process of producing mannitol, pick up 6 hydrogens and remove them. So, you know, the thought is that process of producing mannitol from glucose and fructose is actually depleting the deuterium in the body.
It's quite possible. In fact, I'm really fascinated by the sugars because there are these isomerases, you know, that go back and forth between 2 different sugars. And many of them are actually stripping deuterium off of the molecule, or they're trapping deuterium in one side. It's fascinating. You have some of these sugars that can't be metabolized. I think mannitol actually doesn't really get metabolized, right? Is that
no,
right?
it doesn't. So it just
which is very, very interesting to me, because there's also a cholesterol metabolite that I was really fascinated with that doesn't get metabolized. There's a whole, in the cholesterol synthesis pathway, there's this little side chain that's an isomerase. And so you got these 2 things, and one of them goes on to become cholesterol, and the other one doesn't do it. I couldn't even figure out what happens to it. You know, it may get sequestered in some cardiovascular plaque or something, because I'm wondering if plaque is also a place to sequester deuterium. So I'm starting to think in terms of anytime you have debris accumulating someplace in your body, it might be a deuterium sequester. So you could certainly, if you could get a hold of some of this plaque from a post-mortem or something, you might be able to see how much deuterium is in there, because I would expect it to be high, if my theory is, and that'd be a way to at least do a first step in trying to see if I make any sense with this idea, because it's kind of a wild theory at this point. And
I thought it was interesting, interesting enough to have you talk about it.
I know. Well, thank you for that.
So do we have any other questions? I think we're good with our questions. So
Great.
thank you so much, Stephanie.
That was great. And thank you. And I'm so sorry that I can't be there. I'm sure it was a great conference.
Wish you
It is
could
a
have
great
been here
conference.
with us. It's been kind of interesting.
Yeah. And maybe next time.
Okay, we'll talk to you
All right.
soon.
Thank
Thank
you.
you.
Glad this worked. Bye.